Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So we started immunology, where in first lecture we had an introduction about our immune system, the types of immune system. So we got an idea about what is immunity and how this immunity is devised by an individual. So what are the sophisticated mechanism, the whole machinery that the host has to defend itself against the pathogens. So we learned about the different types of immunity, the innate immunity as well as the adaptive immunity. The innate immunity is one with which we are born, and the adaptive immunity is one which develops over the time in the life in our lifespan. So we learned about the basics of those two immune systems, and then we started discussing about hematopoiesis because the we need to get known about the cells of immune system. So they say all of the cells of immune system, in fact all of the blood cells, they get matured from the hematopoietic stem cells, and the process of maturation, the differentiation of hematopoietic stem cells and into the blood cells, so the mature to forming the mature blood cells, that process is known as hematopoiesis, means blood formation. So we learned about hematopoiesis in detail. So we learned about how the hematopoietic stem cell gets differentiated into different progenitor cells, which are the precursors for the different blood cell types. For example, the erythroid progenitor, this is the precursor for the erythrocytes, means the RBC. So in the blood, we have three main cell types, right? RBC, which are erythrocytes, WBC means the leukocytes, and the platelets, right? So those means thrombocytes. So those three different types of cells, they are in the blood tissue. So out of those three different cell types, WBCs are the ones which are the cells of the immune system. So right, so the WBC, so this is a very old categorization of WBCs or the leukocytes that depending upon whether the cytoplasm of WBC has granules or not, they are categorized into two categories, two types. They are called granulocytes and agranulocytes. The granulocytes are the WBCs, type of WBCs, which have granules in their cytoplasm. So, depending upon the morphology which they further possess or the staining capacities of those granulocytes, they are again divided into different categories. The majority of them are neutrophils, few of them are eosinophils, then the third category is basophils. And there's one more category which is not actually the type found in the blood. It is usually found in the in the tissues, which is responsible for inflammation. But it is also a type of granulocyte. They are referred as mast cells. So mast cells, they have their role in the allergic reaction. So they produce histamines as well as some other inflammatory mediators, so that they can result into inflammation. So those cells are found in the tissue. Okay, so they are not they are found in the interstitial tissue. So we will discuss about mast cell in a separate lecture. So these are not found in blood; these are found in in some other tissues. So for example, the macrophages. There are tissue specific macrophages, right? The microglia that we discussed in neurons, the dust cells that are the alveolar macrophages. Then there are Kupffer cells in the liver. They are found in the sinusoids of the liver. So these are the tissue-specific macrophages. In the same way, we have the immune cells not in our body fluids such as blood and lymph, but we also have them in the tissues, because we need our immune system everywhere, right? So the ones which produce the allergic reaction or the inflammation, and they're found in tissues, those granulocytes are the mast cells. Then the second category is agranulocytes, means those which do not have the granules in them. So they are lymphocytes, okay, and the monocytes. Monocytes, which in future get differentiated into macrophages, so they form macrophages. Okay, so while lymphocytes, they are of two types: B lymphocytes okay, and T lymphocytes, right? So the granulocytes means the mast cell, neutrophils, eosinophils, and basophils, plus natural killer cells, they are the part of our innate immunity. Means they are present in the individual since birth. Okay, so they take part in the innate immune responses, while the agranulocytes, such as the lymphocytes, means the B cells and T cells, they are the part of our adaptive immunity. This does not mean that the lymphocytes are not present since birth. Well, they are present, but in their immature form. So B cells and T cells, they get matured in their respective tissue. So in the bone marrow, B cells are there, and in the thymus, T cells are there. So they get maturated and activated, and then they devise a particular plan to kill that pathogen. So they are adaptive, they are specific, and that is why they are a part of adaptive immunity. The responses that will be seen, that will be given, that will be there due to the B cells and T cells, they will be very specific, and that is why they are part of adaptive immunity. When everything fails, they are our last hope, right? So they are kind of third line of defenses. Well, I did not discuss the line of defenses, so let's discuss about it. So imagine a pathogen entering in your body. So there are multiple ways our body can defend itself against the pathogenic attack. The first priority of our body is to not let any pathogen enter there. So the very first priority is that I don't want any kind of pathogen in my body. But it's not possible, right? So for ensuring that for ensuring the inhibition or avoidance of the entry of pathogen in the body, we have the first line of defense. Okay, so let's discuss about line of defenses here. Okay, the way our body is going to defend itself. So if you have seen the movies, or if you have read the books, whenever you read about a war, so whenever there's a war, the king or the generals, they form a war formation, right? So they decide that what kind of war formation will be formed in the battleground. So the cavalries will be here, the soldiers, for the foot soldiers will be here, the soldiers who are riding elephants and all, they'll be here. So they decide all those factors, and the a whole battle formation is formed. Okay, and this war formation is very important because it is going to provide multiple line of defenses. Because if the first line of defense fails, we still have our second chance, the second line of defense. If the second line of defense also fails, or it gets destroyed, disrupted, then we have third line of defenses. So just this is also a war situation. Defending our body against the pathogen is also a war situation, a war between the immune cells of our body and the pathogen. And so the pathogen is attacking the body. So the body has to defend itself. We are not right now in the attack position. The first and most basic priority of the body is to defend itself. And if the defense does not work, then we attack, right? So the first line of defense. Okay, so line of defenses are three. So the first line of defense is that it is also known as the physiochemical barrier, okay, which is going to ensure that no pathogen enters in the body. So it is a defensive mechanism of the body that no pathogen should enter in the body. So for example, skin. It's the biggest, it's the biggest organ in the humans. So it is going to defend. It is not going to let enter any pathogen inside in our body. So skin, then mucus. Okay, all those they are the part of physiochemical barrier, which is going to ensure the no entry for the pathogen. But pathogens, they are also smart. They also have their own weapons. So sometimes they cross this physiochemical barrier, right? So once they cross this physiochemical barrier, they have to face our second line of defense, which is also known as biological barrier, means our cells. Okay, not just cells, but the biological barrier is various responses such as the inflammation, such as the fever or the antimicrobial peptide that are present in our body fluids. So there are multiple examples of this second line of defense. So well, this is first line of defense, the physiochemical barrier, ensuring no entry of the pathogen in the body. Okay, so they say that no entry for the pathogen. So this is a no entry sign for pathogen, but somehow the pathogen enters. So we need to eliminate it. We need to kill that, and that is why we have the second line of defense, which has the multiple responses. So it is a combo of multiple responses such as inflammation, fever, or we even have antimicrobial peptides, antimicrobial peptides in our body fluids. Then there exists the complement system. Okay, so well, which is also a part of innate system. So these are the different second line of defenses, and we also have the cells, okay, such as phagocytes, which are going to kill the pathogen. So they ensure that if the pathogen has entered in the body, it gets killed wherever it is entering. So the first response of our body against the pathogen will be local response, right? Because wherever pathogen is entering, may it be oral cavity or the urogenital tract or any wound, okay? So if there's a cut, any wound or injury to your body, from that particular part, the pathogen is going to enter, the bacteria or whatever pathogen it is going to enter from that area. So once it enters, the body ensures that the pathogen should be localized there. Okay, so body forms abscess, okay, the clot around that particular part to ensure the localization of the pathogen in that particular part, because that will not let it spread in the overall body, because if it is spreading in overall body, then it will get difficult to tackle that particular pathogen. And that is why first response by our body is to ensure that pathogen is, it is immobilized in that particular part, and that is done due to the inflammatory responses. So first of all, whenever infection is there, the first response that our body is going to give, it will be inflammation. So body has now failed to has failed to defend itself against the entry of pathogen. So now pathogen has entered. So due to the entry of pathogen, the body is going to show the inflammatory response. So due to inflammation, it forms pus, a pocket of pus, which is called as abscess. Okay, abscess. So it is a pocket of pus to ensure that, so abscess, this pocket of pus is formed due to the inflammatory responses, due to inflammatory mediators, and it ensures the immobilization of the, or the confinement of the particular pathogen, the damaged tissue in that particular space where the pus is accumulating. Okay, so what happens is that the inflammation first a localized response will be, it will be given, will be seen, will be observed by our body, and that localized response is inflammation. Inflammation is basically the swelling due to the swelling or pain and heat causing in that particular area due to release of the inflammatory mediators. So we will be having a separate lecture about inflammation, the signature of inflammation, how inflammation is caused. Right now, what you need to learn, what you need to understand is that if the pathogen enters, if the infection is seen, infection is there, then our body ensures the localized response to kill that pathogen. So it will start the inflammatory responses over that localized area where the blood vessels will be dilated, vasodilation will be seen. Okay, and the inflammatory response will be caused, abscess will be formed, a pus pocket will be formed. Okay, but let's say due to some reason the pathogen is not killed in that particular area, our body fails again to restrict that pathogen in that area and to kill that pathogen. So now the pathogen it is going to take over the whole body. Let's say when the whole body has to work against the pathogen, then that global response is fever. Okay, so our whole body heats up, it rises the temperature, and that global response is fever. Then we also have antimicrobial peptides in our, in our body fluids, which are going to have the effect on on the bacteria or the fungi. So whatever microbe is entering in the body, we will have the antimicrobial peptides such as defensins, such as the lysozyme. Okay, so they are going to work over them, so they are also a part of the biological barrier or the second line of defense. Or and in the when the inflammatory response is there, or the pathogen is localized there, or it is damaging the tissue, the phagocytes are going to come, and they are going to do the phagocytosis, means the engulfment of this pathogen, which will end up killing them. So this is also the second line of defense. So you can say that the first line of defense is kind of defensive tactic that body follows, means it is defending itself against the entry of pathogen, while the second line of defense is a kind of attacking tactic followed by the body, means it is going to attack that pathogen because now it has crossed the boundary, it has entered in the body, so it is going to attack that particular pathogen. So this second line of defense, it is, so both first and second line of defense, both of them, they are they are the part of the innate immune response. So they, both of them, they are part of innate immunity, because they, again, see, you can see that they are non-specific, and we possess all of them by birth. We have skin, we have mucous linings, okay, we have the inflammatory responses, we have the antimicrobial peptides flowing in our blood. Our body is capable of giving rise to fever, rising its temperature, and it also has phagocytes. So all of them, they are present since the birth in their, in they are ready to attack the pathogen, they are ready to kill the pathogen whenever there is need. So all of them, they are there since birth, and we need not we need not train them so to attack those particular pathogens. And that is why they are and they are also non-specific, no matter whether it's bacteria, fungi, whether it's virus, or whether it's, whether it's a parasite, unicellular or multicellular, it does not matter to those to those factors, to those barriers. All of them, they are kind of the shields which our body possesses since the birth, which are going to, react and have a response against all of the diverse pathogen. And that is why it is non-specific. In the way the shield is non-specific, right? In the war, shield is going to protect you against, may it be arrow, against against whatever type of weapon your enemy is using, may it be sword, arrow, okay, whatever type of uh, weapon is being used by enemy, the shield is going to do its job of protection, right? So the barrier, the shield, is kind of non-specific. In the same way, the physiochemical barrier means the first line of defense, and the second line of defense, the biological barriers that we have, both of them, they are a part of the innate immunity. So both of them, they belong to innate immunity. While the third line of defense is the adaptive immunity, where now, due to all of them, they failed, the phagocytes, they are failing to kill that pathogen, to engulf that pathogen, the antimicrobial peptides cannot work. So now all of those factors plus the adaptive immunity, they are going to, they are going to work together to kill that pathogen. Now the B cells and T cells will come, they are very trained, and it's like cells, they are our third line of defense. So the cellular and humoral adaptive immunity, they are they are going to work together with the innate immunity to kill the pathogen. This does not mean that if all of those things are failing, means now only B cells and T cells are going to work. No, both innate immune system and the adaptive immune system, both of them, they will work together in coordination to kill that pathogen. So there is always a link between innate immunity and adaptive immunity. So we'll discuss how both of them, they are linked with each other through the complement system and through the cytokines, chemokines. So we will learn about them. So the antimicrobial peptides, the interferons, the cytokines, the chemokines, when they are released, they are going to recruit the cellular parts, cellular components of adaptive system, and the the cells of adaptive immunity, they will also secrete those chemokines, cytokines in response. So this is how innate immunity and adaptive immunity, both of them are linked together, and they are going to work to ensure ensure that the pathogen will be killed. So this was the first line of defense. Second line of defense and third line of defense is adaptive immunity. But this does not mean that adaptive immunity works on its own single-handedly, but it also, this is not the truth. It works with the innate immunity. So there is always link between both of them. So this was kind of, introduction. This is just a start. The factors, they are responsible to innate response, and the factors, the cells, the humoral components that are responsible for adaptive response, they are really, we need to learn about them. They are very complex concepts. So we will be learning all of them in separate lectures. This was not the point of focus. What I want to tell you is that once the innate immune responses starts, so the antimicrobial peptides, they are the humoral component of the innate immune response. Humoral component means anything which is being circulated in our bodily fluids. So that is what we call humoral components. On the contrary, the phagocytes and the cells of immune system, there exist the cells which are the part of innate immune system, and those cells are the granulocytes, and that is why I explained all those stuff to you. So the granulocytes such as neutrophils, eosinophils, basophils and mast cells, plus the natural killer cells which actually belong to the lymphoid lineage, so all of them, they are part of innate immune system. So alongside the phagocytes, they are going to work alongside the antimicrobial peptides. They are going to work to kill the pathogens whenever there's infection. Okay, and while the lymphocytes, they are part of adaptive immunity. So this is very important. So the when the inflammation, when the infection occurs, so whenever the physiochemical barrier has been crossed by the microbe, and now it has infected the body. Once the microbe infects the body, the pathogen infects the body, the first cells that are going to arrive at the site of injury, at the site of infection, they are the neutrophils. So in this lecture, we are going to learn about, first we'll learn about the characteristics of granulocytes, then we will learn about neutrophils in detail. So today we are going to only discuss about neutrophils in a great detail, and then in next lecture, we'll discuss about eosinophils, basophils, plus mast cells. So there will be 2 lectures for granulocytes. Today we are going to only discuss about neutrophils. So as I told you, granulocytes, they are the leukocytes, means the WBCs, the white blood cells. So whenever the white blood cells, they have the granules in their cytoplasm, they are called as granulocytes. So they, depending upon the staining capacities and the morphology of those granulocytes, they are, differentiated into those 4 different types of cells. So all of them, they are multi-lobed cells, and that is why those cells, those cells are known as polymorpho nuclear cells, because the nucleus of those cells, okay, are multi-lobed. So they are having multiple lobes, while on the contrary, the lymphocytes, they have single lobes. So this will be the cell, this is the nucleus, and this is simple round nucleus of the cell. So lymphocytes, they are going to have simple round nucleus. So this will be somehow this will be the structure of a lymphocyte. On the contrary, the granulocytes, they have multiple lobed nucleus, something like this. Okay, let me just draw it here. So this is cell, and the nucleus will be something like this. Okay, so they could have nucleus like this, okay, or nucleus like this. So this kind of nuclear structure, this kind of multi-lobe nucleus will be seen, and that is why, depending upon those morphology and staining process, staining capacities, they are being divided into 3 categories. So the neutrophils, they are called neutrophils because they can be stained by both acidic dye as well as basic dye. And that is why they are referred as neutrophils, because both of them, acidic dye, which is a negatively charged one, and the basic dye, which is the positively charged one, both of them, they can stain it. So so they are somewhat neutral, and that is why they are known as neutrophils. Okay, so the acidic dye such as eosin can stain neutrophils, as well as the basic dye such as methylene blue, it can stain that. And that is why neutrophils are the cells which can be stained by both acidic and basic dye. On the contrary, eosinophils, they can be only stained by the acidic dye. Basophils, they can be Okay, well, I'm not sure. We'll discuss about this in next lecture. So neutrophils are the ones which can be stained by both of the eosin as well as the methylene blue, both of the dyes. So they are the polymorphonuclear cells. Okay. So they are a part of PMN. So they are also most abundant types of cells in so most abundant types of WBC. So the 50% or the 70% of the white blood cells are the neutrophils. So the most abundant WBC is the neutrophil, and they are also predominant in the cell pus. So I told you that the cell pus is formed due to the inflammation. So that pus in that pus formation, we usually found the damaged tissue, and the most prominent component of that pus is the dead neutrophils, or the neutrophils that have that fought, and they are the part of the pus. So we know that in the pus there's the dead immune cells. So neutrophils are the major component of the pus. So the pus is a whitish in color that has neutrophils. So this neutrophils, as I told you, they are the first cells to arrive at the site of injury, because they are, first of all, they are highly phagocytic, and they are very aggressive. That is why, so phagocytosis can be seen not just in neutrophils but even in the eosinophils. So the macrophages are the phagocytic cells, as well as neutrophils are the phagocytic cells. But eosinophils also have somewhat phagocytic characteristics. But the most aggressive phagocytosis which is seen in the cells is what seen by the neutrophils. So the neutrophils are the very aggressive cells, because the phagocytosis that is shown by them is quite aggressive in nature. They are going to kill the pathogens immediately. That is why they are needed they are needed immediately after the infection. That is why they are the first cells to arrive at the site of injury. Okay, they also express PRRs, which are known as pattern recognition receptors. If you don't know about them, please don't worry. We are not going to discuss that here. We will have a separate lecture in the innate immunity, where we will be discussing those stuff in detail. But for now, just remember that neutrophils, they express the pattern recognition receptors, and they are hallmark of acute inflammation. So there are two types of inflammation: acute inflammation and the chronic inflammation. So acute inflammation is severe short-term inflammation. On the contrary, chronic inflammation is not that severe, but the duration is quite long. So if I am to plot them on the graph, acute inflammation will look like something like this. Okay, on the contrary, unlike acute inflammation, the chronic inflammation will somewhat like this. So it is long timed and but mild inflammation. But the acute inflammation is a very it is a very high inflammation in which is short lived. So the hallmark of the acute inflammation is, whenever there's acute inflammation, the cells that needs to work and that are going to be there for sure are the neutrophils. So that is why neutrophils are also known as hallmarks of acute inflammation. Okay, so the granules these are granulocytes. So the granules that neutrophils have, they will be having first, okay, antibiotic substances. So they are going to have antibiotics, proteins, antibiotics molecules, so that they can kill the bacteria, they can kill the microbes. Okay, so the granules that neutrophil possess, they are antibiotic nature. So they are going to have some antibiotics. So I'm going to list them down here. So they will be having defensins. Okay, they will be having ceprosidins. Okay, ceprosidins, all of them, they are antibiotics. Ceprosidins and cathelicidins. The BPIs, which are bacterial permeability inducing proteins. So this protein is going to permeabilize the bacterial membrane, okay, which will be helpful for killing the bacteria. So they are also present in the granules of neutrophils. They also have hydrolyzers. hydrolases, sorry, hydrolases, and they also have lysozymes. So all of them, they are the antibiotic proteins, antibiotic molecules present in the granules. Alongside those antibiotic molecules, the granules of neutrophils have the proteases. Why proteases? Because proteases are really important for tissue remodeling. Okay, because the tissue is going to get damaged, so we need to redesign the tissue first. So for that matter of purpose, we need proteases for tissue remodeling. And once this tissue remodeling is done, so we need to regulate those proteases as well. And that is why alongside proteases, those granules will have protease inhibitors as well. So both proteases and protease inhibitors, both of them, they are important for tissue remodeling. Proteases, they are going to do the tissue remodeling, and for regulating those proteases, we need protease inhibitors. So they are also found in the in the granules of neutrophils. So, and with this, we have the inflammatory mediators such as histamines, which are going to induce the inflammation, and they are going to also cause vasodilation. Okay, so this, they are going to cause vasodilation as well as inflammation. So now, if now you will ask me why the vasodilation is important. So let me tell you, this vasodilation is very important to dilate, to dilate the blood vessels. And why the dilation of blood vessels is important for the inflammation? Because all of those cells, they are in the body fluids, they are in the vessels, in the blood. Okay, so they need to reach from the blood to the interstitial tissue where the infection has been occurred, and we need them to to translocate from the blood vessels to the tissue in such a way that blood vessel does not does not rupture. And for that, the dilation of the blood vessel is important. So what happens is that when the infection occurs, so this is the blood vessel. Okay, so let me just draw the blood vessel. Okay, so this is the blood vessel, and this below it, the underneath it is the underlying tissue, the interstitial tissue. So this blood vessel is lined by the endothelial cells. Okay, so endothelial cells, they are lined up like this. Okay, so what does vasodilation do? It is going to make up space in those endothelial cells. So it is going to, it is going to just make the space between those endothelial cells. So before they were tightly packed, so now a little bit space will be made in between those endothelial cells, through which the cells, which the cells, the immune cells, which are there present in the blood, they can just make their way through those spaces. And that this process of making the way through spaces means, the migration of the neutrophils or the immune cells of our body from the blood vessels to the interstitial tissue, to the underlying tissue, in such a way that the blood vessel does not get ruptured, is known as diapedesis. Okay, and for diapedesis to happen, vasodilation is really important. It is really crucial, because now if the blood vessels have been dilated, now the whatever factors, they whatever cells, immune cells were there in the blood, they can easily pass through those gaps without rupturing the blood vessels, right? So this phenomena of the migration, or movement of the cells from the blood vessels to the tissue without rupturing blood vessels is known as diapedesis. So this is shown by the, this is a characteristic of the neutrophils. But not just neutrophils have the diapedesis quality. Other cells of immune system, they also show diapedesis. So diapedesis is the next characteristics of neutrophils. Okay, so whenever there's infection, first of all, those neutrophils, they are going to show leukocytosis. Okay, and then they are going to show diapedesis. So what is leukocytosis? So leukocytosis is the increase in the number of neutrophils due to infection. So now if there's infection, you need to, whenever there's war situation, the the recruitment of soldier also increases whenever the war situation arises. In the same way, whenever the infection is there in our body, the number of the neutrophils is going to increase, and that phenomena is known as leukocytosis. So leukocytosis and diapedesis, both of them are the characteristics of the neutrophils. So neutrophils, they increase in number, and once they increase in number and the vasodilation is seen, the diapedesis will also be seen. Okay, so this is how these are the characteristics of the neutrophils. Let's now learn about how neutrophils, they are going to, they are going to kill the pathogen. Okay, so we now learn about their functioning, the way neutrophils function. So what happens is that in the neutrophil, so this is a neutrophil. Okay, so inside a neutrophil, I'm drawing what happens when the infection is there. So inside neutrophil, there exists an enzyme known as NADPH oxidase, which works on the NADPH molecules. So NADPH is present there. So on this NADPH works NADPH oxidase. Okay, so this is going to, this is going to oxidize the NADPH to NADP positive. Okay, so this oxidation will release electrons. Okay, so now those electrons, they will, they will react with the oxygen which is present inside the neutrophils to create. So this oxygen, when it reacts with the electron, this is going to form the superoxides. Okay, so this is going to form superoxides, and this superoxide will be then converted into peroxide, the hydrogen peroxide, which will eventually be converted into bleach. Okay, HOCl is the bleach. So this bleach is going to kill the pathogens. Okay, this is going to kill the bacteria. So this is how the neutrophils, they are going to kill the bacteria by production of the reactive oxygen species. So when the what mechanism is devised by neutrophils? So neutrophils, they are going to take help of ROS production, reactive oxygen species production, due to the NADPH oxidase. So the neutrophils uses NADPH oxidase. Okay, so NADPH oxidase, the mechanism. So neutrophils, they can show, they have multiple ways to kill a pathogen. They can do the phagocytosis, means the engulfment, but they are very aggressive because they are going to use reactive oxygen species as well. So the one of the mechanism is the to phagocytose. Okay, so to engulf the pathogen. The other mechanism is to use NADPH oxidase to produce reactive oxygen species. Okay, and that reactive oxygen species is eventually going to kill the bacteria. So one was phagocytosis, other is the use of NADPH oxidase, and the other property that is shown by neutrophils to kill, neutrophils to kill the pathogen is known as netosis. Okay, so netosis means the formation of net. And what is net? Net is the neutrophil extracellular traps. So net literally means net, the net, the mesh. Okay, here the net literally means a net-like formation by the neutrophils. Okay, so netosis means formation of net. Net is the neutrophil extracellular traps, which is a, which is a trap, which is a trap for killing the pathogen. So this trap is formed. Netosis is which we can also call it is a form of cell death. So actually netosis happens due to the death of the neutrophil cells. So once the neutrophil cells, so they kind of sacrifice themselves to kill the pathogen, because once they kill themselves. So what do they do when they kill? So when they kill themselves, whatever DNA is there, they are going to extrude that DNA and they are going to spill their contents. So that spilling of the contents of neutrophil, by forming, and that will result in the formation of net. It is known as netosis. So netosis is a form of cell death. Okay, form of cell death by neutrophils. Okay, by neutrophils, which is going to result into extrusion of their, so they are going to extrude their DNA. Okay, So that is why the whatever net means this neutrophil extracellular trap will be there. It will consist of the chromatin of the neutrophils, okay, the intracellular components of the neutrophils, other intracellular components of the neutrophils. They will also have the the granular proteins which were there in the neutrophils. So those granular proteins have antimicrobial peptides. So that is why it is going to have high concentration, high local concentration of antimicrobial peptides. Okay, so these are the contents, the chromatin of the neutrophil, the intracellular components, the granular proteins, and the antimicrobial peptides that were present in the neutrophils. All of them, they are going to create a trap which is known as NET, neutrophil extracellular trap, which is going to trap the pathogen in that particular formation. And once the pathogen is trapped, it will be killed. So these are the different mechanisms which are devised, which are used by the neutrophil to kill the pathogen. The simply phagocytosis can be done, or it can use. So the major way by which it does the killing is the production of ROS, but in few scenario it also does the netosis. Netosis is a form of cell death of neutrophils. Well, there exists the netosis without the death of neutrophils as well, but there's no clear evidence in vivo for netosis. This has been proved in vitro, but in vivo evidence is not there yet. And if it is there yet, I haven't read the literature so much about netosis, but this is also a way by which neutrophils kills, and this has been seen in vitro conditions. So netosis is seen by the neutrophils in vitro, but in vivo I have no idea about that. So these are the different ways by which the neutrophil is going to kill the pathogen. So we discussed about the characteristics, and we discussed about the way of killing pathogen. So now let's discuss about what will happen if something goes wrong with the neutrophil. Let's say that the NADPH oxidase does not work. So due to some reason, let's say the gene of NADPH oxidase, it mutated, and now it is not non-functional. So once this NADPH oxidase gets mutated and doesn't it's not able to do its function properly in the neutrophil. That will result into no production of reactive oxygen species. And as the reactive oxygen species are not they are not forming, that is going to result into the accumulation of pathogen. Okay, and that accumulation of pathogen will lead to it will lead to lymph node swelling. Okay, it is going to lead to inflammation. So now inflammatory mediators will keep on doing the inflammation, but as the NADPH oxidase is not working, that whole pathogen will get accumulated. It will get piled up in that lymph nodes, because well, the pathogens, whenever infection is there, the pathogens, they are taken in by the lymph nodes, and over this lymph node, the all of the immune cells, they are going to work over on that pathogens, and they are going to kill them. Okay, so lymph node is basically where all of those war is going on. But if that does not occur, okay, if due to some reason, such as the NADPH non-functionality, the pathogen is not getting killed, so it will get accumulated in this particular region, which will lead into lymph node inflammation, and that lymph node inflammation is known as lymphadenitis. Okay, whenever there's suffix itis, is there, it means the inflammation. So lymphadenitis means the inflammation of the lymph nodes. So the non-functionality of NADPH oxidase, it is going to result into lymph node swelling. Okay. Okay. The other serious problems which can arise due to non-functionality of NADPH oxidase is the disease known as CGD, the chronic granulomatous disease, which can also be hereditary. Well, it is a genetic disease. So this is chronic. This could also be genetic, okay? Because let's say that due to hereditary defects, due to some mutation in the gene, so whatever gene was being received by the baby, by the zygote, it is already non-functional. It is already mutated. So that is going to result into the development of CGD, the chronic granulomatous disease, which is a really deadly disease, and it is usually seen in the children, and that is why it is the pediatric patients are usually seen. But if it's not genetic, okay, then it causes due to the frequent infection by the bacteria, or by the the very lethal infections by bacteria and the fungi. So bacteria and fungi, they could be life threatening for children, because, well, our immune system, so this also, we also get infected by those same bacteria and fungi, but they are not that life threatening in our cases, because in the children the immune system is not, if it is not well developed, the exposure to them can cause the severe consequences, the CGD, because the immune system is not well developed in them, and that is why the pediatric patients, they are often seen in this case, in the case of chronic granulomatous disease. So in this chronic granulomatous disease, the NADPH oxidase is non-functional, and that is going to result into defect in ROS production. Okay, so the reactive oxygen species will not be formed, and as they are not being formed, they are going to create the granular structures in the individual who has the chronic granulomatous disease. So the diseases that are there, so the consequences of non-functionality of NADPH oxidase is that swelling or inflammation of lymph nodes, or the chronic granulomatous disease. So this is usually seen in the children. So these are the things which can go wrong. Now, if we have to assess this NADPH oxidase, means you have given this enzyme and you are told that tell me whether this enzyme is functional or not. So for assessing the functional function of the NADPH oxidase, we have a test, okay, and that test is going to tell you whether the NADPH oxidase is working or not, it is functional or not. So NADPH oxidase, for assessing NADPH oxidase function, we have a test, and it is a very simple test which involves only one reagent, and that test is known as NBT test. Okay, so this NBT test means NBT is actually a reagent known as nitroblue triazolium. Okay, so this nitroblue triazolium, this reagent, not triazolium, it's tetrazolium. Okay, my bad, it's tetrazolium. So nitroblue tetrazolium, this reagent is used for assessing NADPH oxidase, and that is why the test is known as NBT test. Okay, so let's see what happens in this NBT test. So it's a really simple test. We take the NBT, this reagent, okay, and in this reagent, this reagent is actually colorless. Okay, so over this reagent, the NADPH oxidase works. So now if the NADPH oxidase, if it is functional, the product that is going to be formed, it will be a blue colored compound. Okay, so if we find the blue colored compound after the addition of NADPH oxidase, means the NADPH oxidase is functional. This means that the individual is normal. Okay, whoever that person is, he he or she has a functional NADPH oxidase. But if the blue colored compound is not formed, this means that there's something wrong with the NADPH oxidase, and that is why that a particular patient is there. So this is how, by using the NBT, this reagent nitroglutatrazolium, we can assess the function of NADPH oxidase. So with this, I am done with the neutrophils lecture. So I have discussed about neutrophils in detail, and well, it was really important to know about them because they are the first ones to react, first ones to arrive at the site of infection. So we discussed about the characteristics that are possessed by the neutrophils. Then we discussed about the ways by which they kill the pathogen, and then we also discussed about diseases related to the neutrophils. If the enzymes that are found in neutrophils they get non-functional, and we also discussed about a test for assessing the function of the NADPH oxidase. So with this, I conclude the lecture here. In next lecture, we are going to learn about other granulocytes, the eosinophils, basophils, and mast cells together. So we are going to have a lecture about all of those granulocytes tomorrow. So till then, see you. Bye bye.